Alkaptonuria, which is also called achronosis since it causes cartilage damage, is another defect in our amino acid metabolism pathway. So here's this larger picture. I don't want you to memorize this. I just want you to realize that our tyrosine, phenylalanine and tyrosine, are eventually going to make its way and get converted into a fumarate through just a series of steps. We're going to focus just on one specific step for the boards, and this step is going to cause alkaptonuria. So let's go ahead and zoom in on that step. I want you to know that the enzyme homogentistic acid oxidase is normally going to convert a homogentistic acid into a mallyl acetoacetic acid. Alkaptonuria is simply a deficient homogentistic acid oxidase enzyme blocking this metabolic reaction. This is going to impair the degradation of tyrosine to fumarate, which leads to a buildup of the pigment forming homogentistic acids in the tissues. What's a major buzzword for alkaptonuria? The most characteristic finding is urine that turns black upon standing, which is why it's given the nickname black urine disease. Other manifestations of alkaptonuria include dark connective tissues, brown pigmented sclera, and debilitating arthralgias, since the buildup of homogentistic acid is going to damage cartilage. Therefore, if you see black urine or black tissues on test day, I want you to think alkaptonuria and homogentistic acid buildup. Homocystinuria literally means an excess of homocysteine in the urine. This is a disease that's going to express locus heterogeneity. Do you remember what locus heterogeneity is? Well, it's when the same phenotype can be caused by several different mutations at different loci. Homocystinuria has three main causes. One, it's going to be a cystathione synthase enzyme deficiency. It can be caused by a decreased affinity for that enzyme for pyridoxal phosphate, which is the active form of vitamin B6, or it could be caused by a homocysteine methyltransferase deficiency. So we have three causes for homocystinuria. That'll be our phenotype, while these could more be represented by our genotype. Treatment is going to depend on the cause. So we have three different treatments that we can implement. Cysteine is made from cystathione. So cysteine is made from cystathione, which in return is made from homocysteine using the enzyme cystathione synthase and also vitamin B6. Therefore, if you're missing this enzyme, or if the cystathione synthase is deficient, then this would lead to a decreased cysteine level in the blood and increased methionine levels. Instead of going this way on the chart, we're now going to go to the left on the chart. So this latter step is going to consume a lot of vitamin B12 and folate. Notice how we're using vitamin B12 and folate to accomplish our conversion of homocysteine into methionine. Therefore, to treat a cystathione synthase deficiency, what you should do is you should decrease your methionine intake. Everything's already kind of being shunted over already, so you'll want to decrease your methionine intake. And also, you're going to want to increase your intake of cysteine because you're not able to actually convert it to cystathione and then later to cysteine. So you'll want to increase your dietary intake of cysteine, also B12 and folate. Since you're shunting everything over here, you're consuming all your B12 and folate, therefore you'll need to replenish it by consuming more on the diet. In the case of decreased affinity for cystathione synthase for the vitamin B6, this is going to be best treated by just increasing your dietary intake of vitamin B6 and also cysteine. If this enzyme cannot grab a hold of that cofactor well enough due to that decreased affinity, if you flood it with vitamin B6 and also intake cysteine, that's going to be the treatment for that. And then lastly, we have a third treatment. If homocysteine methyltransferase, notice the highlighted enzyme here, if this enzyme is deficient, then methionine is going to become an essential amino acid in the diet. So increase the dietary intake of methionine. So three different causes for homocysteine in the urine. Regardless of the etiology, so regardless of one of these three causes, how do patients with homocystinuria present clinically? Patients can present with intellectual disability, osteoporosis, a tall stature, kyphosis, subluxation of the lens, thrombosis, and atherosclerosis. So here we're going to see a dislocated or a subluxation of the lens, just in case if you haven't seen what this looks before. Cystinuria is going to be an autosomal recessive defect in the proximal convoluted tubule or intestinal reabsorption of the basic amino acids, which will include your cysteine, ornithine, lysine, and arginine. I like to remember these amino acids by using the memory aid COLA. I like to take it even a step further and remember it by Coca-Cola, where the first and the last letter of coca is the same as cystinuria. Therefore, cystinuria is going to be your Coca-Cola disease, where you have your four amino acids, your cysteine, ornithine, lysine, and arginine, which will ultimately lead to the excretion of these four amino acids into the urine. So just remember, cystinuria has the defect that leads these four in the urine. One of the most important effects of cystinuria is that cysteine can precipitate and form kidney stones, which are also called cysteine staghorn calculi. What is the difference between cysteine with an EI and cysteine with just an I? I'll show you here. Well, the cysteine with an EI is going to represent the amino acid, whereas the one with just an I is a dimer of the two cysteine amino acids, which are simply connected by a disulfide bond. Patients with cysteine kidney stones may have hexagonal-shaped crystals on urinalysis. The urinary cyanide nitroprusside test is going to be diagnostic for our cystinuria. And lastly, to treat cystinuria, patients can be given acetazolamide or citrate to help alkalinize the urine. Alkalinizing the urine is going to help cleave that disulfide bond in our cysteine molecule to form our two cysteine amino acid molecules. It's also important to drink lots of water and stay well hydrated to help prevent our stone formation. I hope this flash quiz will be an easy one for you. A young boy presents to your clinic with a sweet maple odor to his urine. He's diagnosed with an alpha ketoacid dehydrogenase enzyme deficiency. What amino acids are not being broken down by this patient? Being the Vermont maple syrup connoisseur that you are, you think, I love Vermont maple syrup, representing your isoleucine, leucine, and valine amino acids. So we've just finished going through the key disorders of amino acid metabolism. Let's now transition into the regulation of glycogen. Insulin and glucagon are two important hormones that regulate the synthesis of glycogen in both the fed and the unfed states respectively. Let's now go over the mechanism of each. In the fasting state, the synthesis of both glucagon and epinephrine are going to be driven by low blood glucose levels. Both of these hormones are going to cause the activation of adenylyl cyclase, which synthesizes cyclic AMP, which in turn is going to activate our protein kinase A. Do you remember what the kinase enzymes do? Well, a kinase enzyme is going to phosphorylate its substrate, and in this case, protein kinase A is going to phosphorylate the enzyme glycogen phosphorylase kinase. So we see we're adding a phosphate group here, which is going to activate our glycogen phosphorylase kinase. So this enzyme, which again is a kinase enzyme, can then go on to phosphorylate the enzyme glycogen phosphorylase, which is going to activate it. So notice how we're adding a phosphate group onto it to activate it. The role of the glycogen phosphorylase is to break down glycogen into glucose molecules via a process called glycogenolysis, which increases blood glucose levels to address the fact that you're in an unfed or a hypoglycemic state. So notice how we're liberating free glucose molecules from our glycogen. Remember that glucagon and epinephrine cause the enzyme glycogen phosphorylase kinase to become phosphorylated, which will then lead to the breakdown of our glycogen reserves through the enzyme glycogen phosphorylase. So that was the fasting or the hypoglycemic state. Now on the other hand, what happens in the fed or the hyperglycemic state when blood sugar levels are high? We're going to have insulin be secreted from cells fed or the hyperglycemic state. The insulin is secreted from pancreatic beta cells. This insulin is going to activate a receptor tyrosine kinase, which causes it to dimerize, which sets off an intracellular signaling cascade that results in the activation of two protein phosphatases. So we're going to activate our protein phosphatases. Do you remember what a phosphatase enzyme does? Well, these are going to dephosphorylate both our glycogen phosphorylase kinase and our glycogen phosphorylase. So we're removing that phosphate group from each. So consequently, glycogenolysis is not going to occur, so glucose is going to remain stored inside of cells to prevent hyperglycemia. So we're going to keep our glycogen in its glycogen form. We're going to prevent the breakdown of free glucose. A less high-yield pathway occurs in the muscle in which calcium and calmodulin can activate phosphorylase kinase. So we're going to actually have calcium and calmodulin activate our glycogen phosphorylase kinase. This coordinates glycogenolysis with muscle activity, ensuring that enough glucose is present to make ATP for muscle function. So here's kind of the whole picture now. Here's what it looks like put together. Sometimes it can get confusing if you dive right into this subject with a full picture, so make sure you break it down if you feel overwhelmed. Some general rules are that glucagon and epinephrine result in the phosphorylation of enzymes, we're adding that phosphate group, whereas insulin is going to result in the dephosphorylation of enzymes. These phosphorylated enzymes ultimately lead to glycogen getting broken down to produce our energy. We just talked about how glycogenolysis is regulated, but let's go over what actually happens during both glycogen synthesis and glycogen breakdown. So here's a molecule of glucose. It has 6 carbons labeled 1, 2, 3, 4, 5, and 6. Glycogen is a polymer of glucose, in which the branches are connected via alpha 1,6 bonds, and side linkages off this chain have alpha 1,4 bonds. So here's a glycogen chain being created. Notice how we have multiple glucose molecules linking together. Since we numbered the carbons of our glucose molecule, the 1,6 and 1,4 bonds indicate which carbons are connected. So we have carbons at the 1, 2, 3, 4, 5, and 6 position. So this means that most glucose molecules are connected or linked between the 1 and 4 position, the 1 and 4 carbons. The branches, on the other hand, as you can see right here, are going to be connected via the 1 and 6 carbon position. So here's our 6 carbon, and here's our 1 carbon. So we have 1,4 linkages, and we have 1,6 linkages. As you can see, if we keep connecting with 1,4 linkages, you'll end up with a straight line, whereas the 1,6 linkages are going to come off at an angle, or a branch point. So notice how we're creating a branch. Now, why are we even talking about these linkages? Well, by storing glycogen in a linear form with multiple branch points, long polymers of glucose can be stored in a compact form. So we can see after a lot of work has been done, we get this nice compact form of stored glucose. When glucose levels drop in the body, this stored glycogen form can now get reconverted back into glucose, which can then go on to feed glycolysis and then the TCA cycle for ATP production. Let's go ahead and look here. What is the difference between skeletal muscle and the liver with regards to glycogen? In short, muscles are going to be greedy, and they're going to keep all of that broken down glycogen for themselves, whereas the liver is really generous. The liver is going to make glucose for the whole body. In skeleton muscle, the glycogen is undergoing glycogenolysis, and that's to provide glucose molecules, and therefore ATP, to be used locally in that muscle only during exercise. The liver, on the other hand, is going to undergo glycogenolysis, break down all of its glycogen stores, but it's not really going to be used locally in the liver. It's going to be used to maintain blood glucose throughout the whole body, and therefore it's going to be supplying energy for the whole body during times of low glucose. We'll now demonstrate the mechanism of glycogen synthesis. Where have we seen this glucose 6-phosphate before? Well, hopefully you recognize it from glycolysis, where we have glucose, such as from over here. It's going to make its way over to our glycolysis pathway. That glucose is going to be added to a phosphate group to create a molecule of glucose 6-phosphate, and this will be trapped inside the cell. Now this glucose 6-phosphate is going to be converted into a glucose 1-phosphate by the enzyme phosphoglucomutase enzyme, which is simply going to move the phosphate group from the 6 position to the 1 position on the molecule. Glucose 1-phosphate can then be converted into UDP glucose with the help of the enzyme UDP glucose phosphorylase. This UDP glucose is the substrate or the building block for glycogen synthase enzyme. So here's our glycogen synthase enzyme, and it's going to use these UDP glucose building blocks to synthesize glycogen by adding it to the end of our glycogen chain. Glycogen synthase is going to be activated by glucose 6-phosphate molecules. So if you have a lot of glucose 6-phosphate building blocks, well, you might as well store it as glycogen. So here we see a lot of alpha 1,4 bonds to create a nice long glycogen chain. The body doesn't just want one long inefficient chain though of linked glucose molecules. So what can the body do to increase this storage efficiency? Notice how up here we don't have just one long straight line that's wound up. Well, our solution is going to be in the form of branches. Once that chain has grown to about 10 glucoses long, an enzyme called our branching enzyme is going to take the last few glucoses of the chain, here in red for example, and they're going to move them to a previous 6' spot carbon on that chain. So this will be a 1,6 linkage. Remember I said branch points are always 1,6 linkages. Adding these branch points are going to make this glycogen chain more efficient to store. If you're following along in the book, you'll notice that the figure is going to show the first 3 main steps are going to be used to synthesize glycogen. Now that we've made some nice glycogen, let's break it down. Alright, a lot going on this chart. However, let's break it down. We've already talked about the right hand of this chart. This is all the steps involved in glycogen synthesis. So we've already talked about it. We started out with our glucose molecule, built our glucose 1,6, our glucose 1-phosphate, made our building blocks of UDP glucose, etc., and made our glycogen molecule. Now let's go ahead and focus on the left-hand part of this chart, which is involved in breaking down this glycogen molecule. So the breakdown of glycogen is going to be called glycogenolysis. Our first enzyme that we have to introduce is glycogen phosphorylase. Our glycogen phosphorylase is going to add a phosphate group to a glucose molecule at the very end of our glycogen chain, which is then going to separate it from that glycogen. So here we add a phosphate group, we're going to remove one of the end glucose molecules, and we're going to liberate that glucose. Where have we seen this enzyme glycogen phosphorylase before? Well, it's the same glycogen phosphorylase that we saw earlier, which regulates glycogenolysis along with the enzyme glycogen phosphorylase kinase. So our glycogen phosphorylase is going to sequentially break down 1,4 linkages in the glycogen chain until they get to about a length of just 4 glucoses left. So here we see 4 glucoses in the gray. This 4 glucose structure is called a limit dextrin. So here is going to be our limit dextrin, which glycogen phosphorylase cannot break it down any further. To address this, we need to introduce a new enzyme, 4-alpha D-glucanotransferase, which is part of the debranching enzyme, is going to come in and come to the end of this chain. It's going to cleave off 3 molecules of glucose 1-phosphate from the branch and add it to our end of the chain. And then our alpha 1,6 glucosidase, which is just the other part of the debranching enzyme, is going to swoop in, cleave off this one last molecule of glucose 1-phosphate from the end of the chain. Thanks to our debranching enzyme, our chain is now rearranged, as you can see down here in this figure, which is going to allow our glycogen phosphorylase to continue and resume its function of chopping down these glucoses one at a time off our long glycogen chain. On a smaller side reaction, our glycogen can also be broken down and degraded by lysosomes. This is going to require the enzyme lysosomal alpha 1,4 glucosidase. So over here on this side, kind of this dashed line, which represents our side pathway. This is also known as acid maltase. As the enzyme name suggests, this can only break down apart 1,4 linkages in glycogen, but it's going to slowly convert glycogen down into free glucoses. So another relatively easy flash quiz, what are the end products of glycogenolysis or the breakdown of glycogen? Well, we just covered how glycogen is broken down, but the end product of glycogenolysis in the liver is going to be glucose and free phosphate. If you remember from earlier, the muscles were greedy and they're going to keep all of that glycogen breakdown products themselves. Therefore, muscles are going to break down glycogen into glucose 6-phosphate, which can then enter glycolysis and make some ATP. Since this glucose is phosphorylated, it's not going to be able to leave the muscle cells, so it's not going to be able to freely move throughout the body and provide energy to the body like this glucose can. So this is going to be used locally by the muscle, starting with the glycolysis pathway.